Hi, everybody, and welcome to this VEuPathDB webinar. We're going to get started in a few seconds. There's still some people joining, so we'll let them go ahead and join. I'll go ahead and share my screen so you can see that. 1 second. Okay, I think everybody should see my screen right now. Okay, and in case you were wondering if this is the right webinar that you were planning on joining, so this is the VEuPathDB release 56 webinar. And if you've joined these in the past, you probably know the drill, but we release a new version of our databases about every 2 months. And after that release, we like to have a quick webinar that walks through the different data sets and new functionality. This is obviously recorded, so anybody can come back and take a look at these in the future. But the goal is to have this as a compendium to our release news, which I will show you in a second. For today, we will have 4 presenters. So my name is Omar Harb, and part of the outreach team for VEuPathDB. And I will run through some of the new features in the databases, and then a bit later on during the webinar, I will discuss some of the new data in some of the parasite and host databases, including PlasmoDB, Toxodb, and Hostdb. I'm joined today by Gloria Giraldo Calderon, who is based at the University of Notre Dame, and she will be focusing on new data sets in vector-based. After that, we'll be joined by Suzanne Warnfeldt, who's based at the University of Georgia, and she will be presenting and talking about new data sets in some of our other parasite genomic databases, such as VEuPathDB and Giardia Db and Transcriptome. There's some exciting data sets there. And then finally, we will hear from Evelina Besenko, who's based at the University of Liverpool, and she will tell us about data sets in FungiDB, and so that will be also exciting. And just as a reminder, VEuPathDB is part of the bioinformatic resource centers. There are 2 of them that are funded mainly by the National Institute of Health, and those are the VEuPathDB resource, which supports vectors, eukaryotic pathogens, and host informatics. And then there's another one, BVBRC, which is a bacterial viral bioinformatic resource center, and that supports bacteria and viruses. And I encourage you to visit bvbrc.org and explore their resource. They also have a series of nice webinars and a number of really cool tools, which are exciting to look at. So I'm going to go ahead and switch to browser here. And so obviously all of our stuff that we'll be showing you today will be live demo. One thing before I get started is during the webinar, if you have any questions, please feel free to use the questions panel. All of us on the webinar will be monitoring those, and we will answer them either directly in the chat, or we will answer them verbally. Feel free to ask any questions, so you can ask questions about what we're describing at the moment, or if you're curious about how to do a certain thing, ask the question here, and if we have time, we will love to demo it and show you how to do it, assuming you can do it in our resources. So, I'm looking here at the VEuPathDB main website. And one place, or the place where you can get information about what's new in the latest releases, is available right here in the news and tweet section. So you can click on this section right here. This will expand the news and tweet section. And you will notice here that there are a number of news items listed here. You can peruse them here, or you can go and see all of the news. And you'll notice, for example, the top news item here is about a tick and tick-borne pathogen open community webinar series. And so this is, again, if you're interested in ticks and tick-borne diseases, I would encourage you to visit this page for this webinar series and register for these. There'll be about 6 or 8 webinars, and they should be very exciting, and a place for you to learn more about what's available in these resources, and also to let us know what you need and what you like to see in these resources. The news section includes then news release items for each of our resources, and so you can go through them and then see what is new in each one. And obviously you can go and visit any of our independent sites and visit them and visit the news section there, and you can look at news that is specific, for example, to PlasmoDB. Okay, so I'm going to cover quickly a couple of the new features that are in VEuPathDB, and all of these resources, all of these new features. The cool thing, since all of our databases are built on the same infrastructure, all of these features that we develop are available for all of the resources. So it doesn't matter which database we're demonstrating, that feature will be available for you in your database of choice. And so the first feature is Multiblast. So you may have noticed that if you use Blast frequently, for a while we had 2 Blast options. One was an old Blast, which was a single Blast option, and then we introduced Multiblast as a tool. A few releases ago, it was available as a beta tool. As people got used to it, and we made sure it's functioning appropriately and performing well. And so now, if you go here, you'll only have this Blast tool it's a Blast tool. You can do a single sequence, but it's multi-Blast capable. And what's cool about this is that you can go here and you can paste a FASTA file. So I'm just going to go to this sequence, a bunch of sequences. So I'm going to copy these, go back here and paste them. So while I'm doing this, and these are, in this case, I'm looking at sequences from a Toxoplasma file. So I wanna blast these against I'm gonna just pick a Toxoplasma genome. But what you'll notice here is, as I'm doing this, I'm trying to find this one Toxoplasma strain. Nothing is coming up, and this is obviously, I'm like, well, wait a minute. I know Toxoplasma is part of the VEuPathDB resource, but hopefully everybody noticed this little icon here on the right. I had forgotten that, the last time I was here, I changed my organism preferences. So this is a new feature. So I'm giving a plug here for a second feature that has been available for a couple of releases, but just to remind you that this feature exists where you can go in and narrow down which organisms you would like to search and query in the database. And this, again, functions on all of our resources. So for example, if you're in FungiDB, which has many organisms, and you may be only interested in Ascomycota, for example, then you can limit your organisms that you search. And this obviously improves performance, but also it allows you to, it takes away sort of all the other organisms that maybe you're not interested in seeing in your results or in the filters and such. So what I'm going to do, instead of changing my organism preferences right now, I'm just going to go back to my search, and I want to limit to Toxoplasma, but it's not part of my list here. So all I'm going to do is I'm going to disable this filter at the top here, and you'll notice now everything is available to me. And now I'm going to go to Toxoplasma ME49. So I'm just filtering this table. I've selected ME49, and now I'm going to paste my list of sequences, and I can run my job. Okay, and once you get your results, you will get this combined result list. And so these are all the blast hits. You can go also to the individual results if you want. And one new feature that we added in this release for Multiblast is a new download option, which is you can download this table as a CSV or basically a delimited file that you can open in Excel. You can import this into Excel. And so that's nice. So this was a request from a user, and I want to give a plug here to making sure you use our Contact Us link and send us requests. So if there is a tool that you feel like can be improved by adding a new feature, or there is a feature that is missing that you think would be useful for the community and yourself, obviously, let us know about it, and we'll put it on our list of things to develop. And of course, at some point it'll become available and hopefully make your life easier. So before I switch it over to Gloria, I'm going to quickly show you another feature we added in this release, and that was a direct request again from a community member, and that is in the way we colored the UTRs and introns on our gene pages in the sequence section. So here, I'm just jumping to Toxodb. I'm looking at I just picked a gene. Actually, I picked a gene from my BLAST results in the previous page, and I went to Toxodb to this gene. And as you scroll down on the gene page, you'll notice here that it has, this is the gene here. It's going from right to left on the right to left strand, basically, or the reverse strand. This gene has multiple exons, as you can see here. These blocks are all exons, they're introns. It also has these blank spots here, the white spots here are the, at the ends are the UTRs. And then let's say you really want to, you just want to go look at the sequence. So I'm just going to go down to the menu here. And sometimes, this is a very long menu. The nice thing is we have these menu filtering all over the place, so I can just start typing sequence. And then you'll see here there are different options that contain the word sequence in them, but here's one that contains a sequence. So I'm just going to go to the sequence section. Here's the sequence section. I'm going to expand this, and I can get the predicted protein sequence. I can get the mRNA, which includes the UTR. As you can see here, it's highlighted in pink. And now if I go down to the genomic sequence and I expand this section, you will notice here this was a multi-exon gene. It has a UTR highlighted in pink, the 5' UTR. The exons are just white. And then you'll notice here all the introns in your sequence. In the past, these all used to be light blue, which was similar to the highlighting. So if I try and copy something, it was essentially the same highlighting, which was bothering a bunch of users who weren't able to easily copy stuff. And that was legitimate and was an easy fix for us, and we're able to implement this immediately. And so I wanted to make sure we highlight this and highlight how communicating with us is quite helpful. Okay, so I think without further ado, I'm going to go ahead and switch this over to Gloria. And Gloria, I'm going to make you the presenter.
Thank you, Omar.
And, yeah, and again, as I mentioned, feel free to ask questions in the question pane, and we will obviously answer them throughout the webinar.
Okay, can you see my screen now?
Yes, looks good.
Great, thank you. Hello, everybody. So as Omar said, I'm going to demo what we have new in VectorBase. As he just showed you, when you come to the home page, click here on News and Tweets, and we are going to see what we have new for release 56. So for release 56, we have new data from both the Omix and for population biology. For the population biology, we usually divide this in two sections. One is the new projects. In this case, we have 8. And also some projects that we might have some issues and we are fixing. So I'm not going to demo this part, just the ones that are truly legitimately new projects. So you will notice that each one of these bullets is the new project. And we have this unique accession number from VectorBase project, and the number, the consecutive number that identifies this project. Each one of these projects has 2 links: dataset info, which is the metadata corresponding to this project, and view on map. And then on the map, you will only see this data set. So for example, this is the page that we released these data set pages for the map data. We're available for the Omix data set, but not for the map data in the last release. So we already show you how that looks. And this is the other link, view on map. So notice it's only for this VectorBase project that we are showing the data, and it's telling me that this is population abundance data. And if I want to see what this data is about, remember you can interact with the map, zoom in, zoom out. I can click on this link that looks like a table, and this list of details is giving me all the how was the data collected, how was it processed, the sex of the samples, or anything that I want to know. If I delete the filter, so I will see all the data that we have in the map and the different categories, and you can, come back to the different filters, only population abundance data or insecticide data, and interact with the map. Okay, so what data sets we have here? So we have new for this release, we have population abundance, insecticide resistance, genotypes and phenotypes, and infection status data. For the and if you are not coming from the release page and you want to see the population data from any of our pages, you come to Tools and you click on Map View, and that will load the map for you, and you can start interacting with the different samples or typing, you know, any type of query that you want. Okay, so for the Omix data sets now, for the Omix data sets, we have genome sequence and annotation. And actually, the first 3 ones we already had released in VectorBase. Aedes albopictus C636, Anopheles arabiensis dongola 2021, and Anopheles coluzi mopti. And I can see in the list of attendees, we have some of the people that submitted this data to VectorBase. So we already had the assembly in a previous release, but we didn't have the annotation. So now the new thing is that now we have the annotation. And the difference that makes to you is that now you can ask more questions. So in addition to only have the chromosomes or the super context available for you, now you have the genes to ask specific questions. For example, if you have RNA seq data, which I will demo in a second. There is a 4th genome that is completely new. It's a new Culex quinquefasciatus Johannesburg genome. And this one, we don't have a publication yet, just the bio project, and we'll update that once the data providers let us know about that. One thing about these 4 new genomes is that all of them, and I'm going to show you how that looks, have the gene IDs in the vector-based format, and we also demoed that in the previous release, and here's an example of how that looks, what the letters mean, the consecutive numbers, that the R means that we are looking at a transcript, and the P means that we are looking at a peptide. So all these new 4 genomes, the gene IDs come in that format. So if I click here, I will see all the metadata for this genome. But if you are not coming from the release notes, you can always come to Data, Organisms, and here you will have the list of all the genomes. You come, for example, you want to see C636, which is one of the new genomes. Here you click here, you will just see the metadata page that I was just showing you. And if you click on genes, a strategy will start for you. And here we'll see all the genes for that genome. And this is what I was telling you. This is they are in the format, the gene ID format is the vector-based standard format for the genomes. Okay, something special about this Culex genome is that for the next release, and we have a note here, is that this strain will be changed. So currently it's just now Johannesburg, but with the abbreviation of the 3 letters, but it will be named Johannesburg 2020, so you don't confuse this with the other Culex genome. I'm going to show you. So if I look at only the Culex genomes, so normally in the literature, the Johannesburg sometimes is abbreviated just with these 3 letters. So to avoid any confusion, this, the newer one, that's why in the next release it will be changed. The last thing I'm going to show you is that we have this RNA seq data set. So as we have shown in previous webinars, you can start data mining the data sets we have from searches, for example, here, and you say, oh, I want to look at RNA seq data sets and start from there. But you can also start from the specific data set that you want to query. And if you're not coming from this page here, you can come from data sets in VectorBase. And for example, we currently have 1,218 data sets, and this includes both population biology and the omics data sets. So for example, I'm looking at midguts and salivary glands. So this is midguts. And so notice how it starts to filter, and now out of all the data sets, I am only looking at 26 because I'm using this as a keyword. So from there, you will start your search, you will look at the data set. And if I click here in the data set, either starting from here, from that data set page, which searches are available? So I have available full change, percentile, and I want to do, for example, a fold change. So for this data set, I want to know for all the protein coding genes that are up or down regulated with at least a fold change, of 2 or more, what is I want to see the expression value that I have between our reference samples and comparisons. And I can select anything that I want. So for example, and this was selected because I looked at this before, so I can reset the values if I want. So I want to compare my midgut samples against my salivary gland samples. And I want at least for each transcript to have 10 reads. And that's what this floor means. And I start my query. And this is the one that we ran before, which I was showing you the genes for the new genome, but notice now is gray. But the one that I just ran for you turns yellow. And here I have my midgut and salivary gland genes. And because these samples were given an infected blood meal, so I'm expecting some genes related with digestion, with the immune system, and that's what I see here. And I can add more steps, and we have demoed this before. So in this case, I'm going to combine this sample with, for example, signal peptide genes I want to see, and we are looking at Anopheles gambia pest, so I select that. I am just going to finish so I don't run over the next person. So the last thing I want to say is that, so these are all the genes that are have differential expression from the data set we just selected for the parameters we selected. And these are all the genes in the genome with signal peptide. But the intersection between these two data sets is 1,000 genes, and that's what we have here. And we can keep growing our strategy. And notice this is highlighted in yellow. I can change what I highlight. And depending on what I highlight, that's what I will have here in this table. And these are the data that I can download. So that's all that we have for Vectorbase. I don't know if there is any question or we move over to the next person. I don't know, Suzanne or Eve?
Yeah, I think it's Suzanne. So let's go ahead and move on to you, Suzanne. I can make you the presenter. You're already the presenter. Okay, perfect.
Okay. Do you see the homepage of TriTripDB?
Yes.
Yes, and we can hear you well.
Yeah, that's great. All right, so I'm going to just talk about the data that we added for 56 in TriTripDB, Crypto, Giardia, and AmoebaDB. So TriTripDB is first, and I'll go to the news. And we see that we added one new genome sequence with annotation, 3 RNA Seq data sets, and we've updated functional annotation for 35 genes on TB927. So the annotated genome that we integrated is T-Brucei EARTO, and it was sequenced with PacBio methods and assembled de novo. Annotation was accomplished with Companion, and they used transcriptomics to guide the annotation. From a biological standpoint, this genome is interesting because it's a lab strain that is pleomorphic in mammals and fly transmissible, and those are properties that most lab strains have lost. So this could be really interesting for people to look at. The first RNA Seq data set is a study of the effect of heat shock on Promascot transcriptome. Here's a sample graph of the data. There were 2 samples taken in this experiment, one with Promascots under normal growth conditions at 26 and one under heat shock conditions at 37. So you can search that data, you can look at individual gene pages, and you can go to JBrowse and view that data along aligned to the genome. Those are the 3 ways that you can look at this data. Our second data set that we integrated was the life stage transcriptomes, and that had 3 samples, mastigotes, metacyclics, and procyclics. I have a sample graph here. And this experiment, the experimental design included biological replicates, so we were able to do a statistical analysis on this, and we offer that in our searches and and in our display. Our last RNA seq in TriTripDB was a Promascot transcriptome of this Endotrypanum species. It's just one sample, so no differential expression here, but these descriptive style experiments are important. You can use the search to find genes expressed in this Endotrypanum Promascot, and you can use it in our search strategy system to restrict a set of genes by those expressed in the Promascot. You can also use our orthology transform function to investigate this further. If you are interested in the functional annotation, there's a Google Doc linked here that summarizes what was done in TB927 in release 56, and you can also download all of those changes from GeneDB. Okay, I will move on to CryptoDB and go to the news there. Oh, you know, I wanted to show you one other little trick in TriTripDB. So if I, and it's about the RNA sequence data, if I go to searches to the page that Gloria pointed out, RNA seq evidence page, I'm presented with a list of all of the RNA seq data within TriTripDB. And if I type 56, that narrows the list down to all of the new data. You might get some others because in the descriptions for these data sets, there might be the number 56, but in this case not. But it's a great, easy way to look at and then use these searches to access that data. Okay, on to CryptoDB. We have one new population data set, the C. hominis isolates from 4 African locations. This includes whole genome sequencing of 28 isolates. And without going into too much detail, I'd like to show you where to access that. So I am in the searches. I've opened up SNPs, and all of these searches are available for this population data set. And I'm going to choose differences within a group of isolates. I'll make sure that I've chosen C. hominis because that's what these isolates are, and that's what we've aligned it to. And then the second parameter here is for choosing your group. And I can click on data set and then choose just this one data set. And I see that I've now restricted this search from 81 samples to 28 samples. And I can use this metadata over here to restrict it further. If I open geographic location and then country, I can say I only want to search these isolates from Tanzania. So that's our population data in CryptoDB. I'll move on to AmoebaDB. And we see that we have added 2 assemblies, Entamoeba histolytica. These are good for blasting against. And we've added an RNA sequence data set for auranophin adapted trophozoites. So there are 2 samples here, wild type versus resistant. And the authors collected biological replicates, so there is a statistical analysis available for these. And this data can be used to interrogate, the mechanism of resistance for this Entamoeba species. It can also be added into our search strategy system, and the orthology profiles are available there too. And last but not least, Giardia Db. I'll go to the news there. And we see that we have added 1 genome sequence with annotation. It is a new version of Giardia assemblage A, isolate WB. And to distinguish it from the version that's already in Giardia DB, we've added the year, we've indicated the year of its upload into NCBI. I'll show you also, we can go to the organisms page. And if I type in WB, I can restrict this to a few organisms. And I see the first one is Giardia assemblage isolate WB, and the second one is the newest one that we've integrated, Giardia assemblage A isolate WB 2019. Now, this version of the genome is important because it's resequenced. It's a whole entire resequence of the genome with PacBio methods, and it's assembled to near complete genomes. So if you are interested in this, it's a great new addition to our sites. Please check it out. And if you are interested for more information, or if you would like to tell us perhaps this should be our internal reference for assemblage AWB, please use this contact us form and let us know about it. That's all I have.
Great. Thank you, Suzanne. I'll go ahead and switch over to me so I can wrap up the parasite databases. And what I'm going to do is, before I get into a couple of the things I was going to demo, there was a question about what do we mean by non-unique RNA sequence data, right? And so if you look at any of our graphs or any of our genome browser tracks that show the coverage of RNA sequence data, we always have unique and non-unique tracks. And that could be confusing, but really it's pretty straightforward. So when you do RNA sequencing and you map the reads back to the genome, some of the reads are going to map equally well to multiple locations. For example, if you have repeat elements, right? Or if you have a gene family that's very closely related, and often they have a big portion of their genomic sequence is identical. And so as a result, a read can map to more than one location. And the question is, well, what do you do with that read? In many cases, people just throw these out. They don't even factor them in the calculation, in any calculation analysis of gene expression. And we do the same thing, but what we also do is we give you an indication as to whether a particular gene had a lot of non-unique reads aligned. And that's important because it could help you, because sometimes a gene may look like, oh, this is not expressed, or it is highly expressed, either way. But then you look at the non-unique reads and you're like, well, this gene has a lot of non-unique reads, so I can't be 100% certain that this specific gene was the one that's upregulated, or possibly a gene could show up as it's not regulated, but it's mostly non-unique reads, right? So it could be potentially expressed highly. It's just that the algorithm cannot determine where the expression is coming from. And so I'm showing you here a genome browser view in Toxodb. You can do this in any of our databases that has a RNA sequence data, which is available. In this case, I loaded some tracks for an RNA sequence data from Adrian Hell's group, which includes forward and reverse strands. So this was a strand-specific data set. And for each one, I loaded the non-unique and the unique reads. And I'm looking here at a gene where everything is unique, so you don't see anything in the non-unique reads. But if in Toxodb, if I go, for example, I'm just going to go to the search box up here. I'm going to type the word SAG, and I'm just going to pick one of these SAG genes. These are surface antigen genes. It's a whole family. And now you'll notice, for example, here's a SAG gene. It's a family of genes that are highly identical to each other. And you'll notice here there's a huge peak in expression in the non-unique section. Even though there is some unique reads here and can be used potentially for determining if this particular SAG gene is differentially regulated, there's a lot of non-unique reads that we don't know where they're coming from. Do they belong to this transcript, or do they belong to one of the other, whatever, 30 or so SAG genes that are available? Okay, so trying to see. So in terms of, there's a question about quality control from Maria Tuz, and so the answer is we don't do cracking here. We're basically, and we don't necessarily quality control the RNA sequence data in the sense that we don't go through FastQC, for example, and say, oh, the quality of these reads are not good enough, and we're not going to load the data. We load the data, and one thing we do is, if you remember, Gloria showed an RNA sequence query, and there was this floor which you can change in terms of how many reads per gene are used to determine the differential expression. And so that gives you an indication of what the coverage is like for a particular RNA-seq experiment. And so the higher the reads have to be in order to determine differential expression, the potentially the lower coverage there is from a particular data set. But we don't necessarily quality control the reads themselves. Okay. Hopefully that answered your question, but please ask more if that did not answer your question. So I'm going to quickly just give you a quick tour of a couple of data sets that are new in Plasmodb and in Toxodb and in Hostdb. I'm not going to run through most of the queries just because they're going to be the same as both Gloria and Suzanne showed you so far, and Eve is probably going to demo a few of those as well, so I won't show you another RNA sequence one most likely. But I did want to highlight that in Plasmodb, in the latest release, we added a phenotype data set, which is shown here. And so this is coming from John Adams' group in University of Florida in Tampa, and where they've been doing piggyback transposon mutagenesis of the Plasmodium genome. We already have a nice data set from them in Plasmodb of a whole genome mutagenesis and then assaying growth fitness. In this case now, they're going back to the same libraries, or maybe perhaps some new libraries, and they are determining which genes affect different phenotypes. So in this case, we got the first sample of data set of genes that are affected by heat shock, and there will be many more of these coming in. So if you're interested in this type of data, I would encourage you to explore this experiment. I'm going to go back to the home page because I want to show you that these searches we have. So I'm just here on the left-hand side. We have all of our available searches in the database, which are many, and sometimes you could open them and try and navigate to a particular search of interest. But I think the easiest thing is to use this filter. So hopefully you'll notice that all of our searches have these filters on them, which allow you to find things. And in this case, I want to look for phenotype. So I just started typing phenotype, and you notice here there's a whole section on phenotype evidence. And you can look in your database of choice. If there is phenotype data, you can see if there are any searches there. And you'll see here that we have different types of searches of phenotype searches in Plasmodb, including this new one, which was the heat shock response one. They have different types of searches depending on the type of phenotype data. This one in particular is you can basically limit the genes based on the response they had from the phenotypes that they assayed. So I can go in and say I want to take anything that had heat shock response in this range, and you can look in the data description to find out what these mean, and I can maybe reduce it. And you can see here I'm getting a real-time view of how many genes I'm going to get back as I filter these. So you can do this. You can also jump around between these filters, and it'll show you basically of this subset, what do they look like, or where do they fall in these other filters that are available to you. And ultimately, when you're happy with what you found, you can just get your answer, and this will return those genes that met the criteria, and then you can explore them further. We also have information on the gene page for these phenotypes. And so there's a phenotype section on the gene page. And again, right here, I did this before, but I filtered by phenotype. And I'm going to go to the phenotype graphs. And you'll notice here that we have a graph for, depending obviously on the experiment, if it makes sense to put it in a graph, but here's a graph for the entire whole genome transposon mutagenesis. And you can see that this gene is considered dispensable. In other words, when it was mutated, it did not really affect fitness of the organism, so in the stage that was assayed. But we also have a phenotype score table. In this case, it includes not only the fitness scores from this experiment up here, but also the fitness scores from the heat shock experiment that I just showed you. And this table will expand. We'll also be adding more graphs here. And again, I encourage you, as Suzanne did, if you know of data sets for whole genome phenotype assays for your particular organism of choice, let us know. We'd love to make sure we include them. We already have many, but definitely there's always room to add more. I'm going to jump quickly to Hostdb. And so in case you're not aware, we have a database called Hostdb, which the goal of this database is to integrate host response to pathogen infection data. And obviously we're looking here at eukaryotic pathogens in this case. And so for this particular release, we added 2 transcriptomic data sets. Both of them are related to Plasmodium. One of them looked at the response to bronchial epithelial cells after stimulation with hemazoin. So as you know, Plasmodium generates a lot of hemazoin, and that could have an effect on respiration. And the second one was human brain transcriptome, and that was in response to parasites expressing a particular PISC2 protein. And so, and again, as everybody indicated so far, each of these is well described in our dataset page. And so you can go here, you can get access to the paper, so you can learn more about it. We have some information here. You can see who the contact person is, where this data came from, the searches, and also some of the, an example graph of a gene from this experiment. So you can take a look and see what to expect when you run your search in terms of type of data. And so, but I'm not going to run these searches in specifically because it's going to be exactly the same as you saw demoed by the other resources. And then finally, just to highlight in Toxodb, we have a data set which looked at again, this is an RNA Seq data set. So again, I will maybe quickly highlight it just so you see it again, but this is a data set looking at the response of toxoplasma after treatment with tartralone, which is a anti-apicomplexan compound. This is coming from, let's see, I forgot who it's coming from, but what we can do easily is look at the publication, and then you'll see here, oh, this is from Roberto O'Connor's group. Yeah. And so, yeah. So, and so as you go down here, again, you can run the searches that are available for you. And so I'll just pick one of those. So here, this is a fold change experiment or search. I ran this just before the call just to make sure everything is working. And you can see here what parameters I searched. I looked for any gene that is downregulated by at least 4-fold when I compare the samples that were treated with 24.2 nanomolar of the compound at these 3 time points, 4, 8, and 12 hours, compared to the controls for 8 and 12 hours. And I ran the search. I'm not going to run it again because it's in my strategies already. This came up with 12 genes that are downregulated on average compared to the other samples. And if I go here to the right, you'll see here we get these graphs which show you the expression levels, and you can confirm this. You can see here that, oh yeah, it is lower here than the controls. And then you can go through all of these and explore them, and then maybe pick one of the genes specifically that you want to look at in more detail. And before I turn it over to Eve, just to point out that we do have a lot of analyses tools that you can run on your results. For example, you can run Go enrichment or metabolic pathway enrichment or even text enrichment on your results if you want to try and find connections. Okay, so I will go ahead and switch this over to Eve.
Amar, just a quick, before you switch, there's a question about UniPro IDs. Would you like to answer it now or after the...
I can look at it. So the question is, especially for Transcriptome, gene names into UniProt IDs. Some genes have multiple matches and some have none. Right, so basically so obviously we're not generating the UniProt IDs. We have a version of the genome that is mapped to UniProt. And it is possible, if I understood the question correctly, it is possible, depending on version numbers, that a gene did not maybe maps, or maybe a gene got removed from a current annotation, and then as a result, you won't find that UniProt ID, or a gene got split into 2, and then it matches that. And there are probably other nuances to this which I'm not describing appropriately here, but that could be the issue. So you could have cases where you have one to many or many to one mapping between our gene IDs and any other gene IDs out there. So even GenBank IDs, because again, there's a lot of versioning that happens, and depending on the version of the genome, that could cause issues like that. And I know, I think Uli, who's one of our main curators on the group, I think maybe listening in. So Uli, if you have additions to add to this, maybe you can put them in the chat or as a question, and then people can read it if you send it to all. But that is at least as far as I know in terms of UniProt IDs. Cool. All right. All yours, Eve.
All right, thank you very much. So you should be seeing the screen on FindGDB release 56 news.
Perfect.
Excellent, thank you. And I will start by walking through the updates, starting with genome sequences and annotation updates. This is, I want to mention that we have a first update that have been where the genome has been updated through Apollo, which is a web-based genomic annotation editing platform. And if you're interested in improving the quality of the genome, structural or functional annotation of the genome that you're working on, we have this tool currently available on all genomic sites. The tool is available under the Tools menu, where you can click on the Tools, navigate to Apollo, and that is the landing screen that you will see. So we have a lot of tutorials and even a webinar previously recorded to get you started if you have never worked in the system. And just quickly to show you what you may see when you try to work in Apollo, I'll show you the screen for Cryptococcus neoformans. And this is very simplistic view. And what I have here is essentially annotated transcripts, also RNA seq coverage, which is an RNA seq evidence from a strain-specific data set that provides supports for the current gene models. And what I have done, I simply double-clicked and moved this gene upwards to move it into user-create annotations, which is the space where you can work and adjust UTRs or exons, create new genes, and adjust various coordinates. And this work can be saved, and also once you complete your tracks, they will become visible to the rest of the community within the 24 hours on FindGDB and other genomics site. And deleting annotations that you may not like, or if they're not correct, you can simply double click to select them and delete them from the tracks. So the system, there is a delay a little bit on my side. So the system is quite easy to work in, and if you have any questions, you can always send us questions via contact us email. But to come back to FindGDB, the first genome that has been annotated via Apollo is Trichodorma rhizii, where we have created and integrated 225 changes that include various modifications that consist of adding new genes or creating modifications within existing gene models. And if you're interested in learning more about the specific modifications made, click on Break Down the Changes, and you'll be navigated to a Google Doc that has a summary of all changes. Now, we have also added new genomes, such as genome sequence and annotation and just assemblies. And these genomes sort of fall in the category of various pathogens, such as phytopathogens, animal or human pathogens. We also added a new high-quality genome for Aspergillus flavus. And notice that this genome has an addition of 2020. That's to differentiate from an older genome assembly for Aspergillus flavus that we also have in FindGDB. The difference between these two is that the 2020 genome is actually a resequencing and de novo assembly of Aspergillus flavus. And this is a hybrid assembly that was used using nanopore and PacBio technologies. And this is also gapless assembly that contains 8 chromosomes, where 7 out of 8 chromosomes are completed from telomere to telomere. And we also have, as I mentioned, various pathogens and excuse me, for the genomes for which we just loaded the assembly sequence, such as Rafaela loricola, we will load the annotation in the subsequent release. And as Gloria mentioned before, having genome annotation is quite important, as it makes possible for us to activate gene-centric searches. And for example, if I click on the dataset record page for Trichophyton rubrum, which is a dermatophyte, and scroll down to the bottom of the dataset record page, you will see that there are a list of searches that are available for this genome. And when you have the annotation available, you can actually run various gene-centric searches. Now, coming back to the release, I also want to mention that we have a variety of SNPs, SNP and transcriptomics data loaded in this release as well. A lot of these data sets cover the subject of drug evolution in human and animal pathogens. And we have several data sets that are SNP data sets that have been loaded for established and also emerging pathogens, such as Candida auris. And here's, for example, one data set where samples, which are the isolates, have been evolved through the exposure to azole antifungal drugs. And if you navigate to the dataset record page, I won't run the analysis. You will notice that this particular dataset is associated with several types of searches that include where you can identify genes based on their characteristics, such as you can identify non-synonymous SNPs. As we know, they may contribute to drug evolution in pathogens, or you can actually look at the copy number variation on the chromosomal level, as we know that these type of events are the driving force behind quick evolution, again, of pathogens within clinical settings. And so, for example, if you click on the SNP characteristics search, you'll be redirected to a page that will look something like this. And because we have multiple genomes and various data sets loading for them, you'll need to first identify Candida albicans as the organism. That will also refresh the list to update the number of available data sets. And the data sets that I just told you about, this is the Candida albicans adaptation to azole and antifungal drugs. So you can click on this data set, scroll down to select your parameters. And in this case, you say, well, I want to identify all non-synonymous SNPs. In this case, at least one or more. And for just simplicity, I will leave all the selections at default and click Get Answer. And so this is essentially a very quick and easy way to create a first step in a strategy, your search strategy that identifies non-synonymous SNPs. And you, as Amar shown before, you can deploy various types of additional analysis to mine the data further. Now, coming back to the news, and I want to also mention that we loaded quite a few of functional data sets, and that's include RNA seq analysis data for various pathogens, Candida, Coccidioides, Cryptococcus, and also companion data set for Saccharomyces cerevisiae, where both Cryptococcus, not Cryptococcus, but Candida glabrata and Saccharomyces cerevisiae were investigated under the same conditions. But returning back to the strategies, as I mentioned, you can create strategies in FindGDB using various data sets. And let's say we want to actually take a closer look at the Candida albicans data sets that we have loaded in this release. And let's take a look at the calcin data. So what calcin is, this data set actually was generated from a large scale analysis. And the authors have used large scale RNA sequencing studies that are available at NCBI. To be exact, it's about 853 RNA sequencing runs from 18 large scale data sets. And they essentially analyzed and generated a network of co-expressed genes to predict their function. So using this data set, you can mine the co-expression data essentially on the whole genome of Canada. So let's go ahead and add a step to the strategy that I just set up and ask a question of how many of these non-synonymous genes in Candida albicans that were undergoing drug evolution in lab actually have co-expressed genes based on the calcin network. And so what I can do, I can click on that step, and because we're still searching for genes, my selection will be the same. I will navigate to the RNA Seq evidence because we're looking at the transcriptomics data, and search for calcin. And so when I clicked on this data set, a parameter screen will be returned, so where you can either, if you're just curious, search a default setting. This is just an option of a few genes if you like, or if you have your own genes, you can also replace this with that list. For just simplicity, I will leave everything at default. And by the way, if you're ever curious to, if you want to learn more about the specific conditions used in this experiment, you can always click and open the link in the new tab that will bring up the dataset record page. But in this case, I'll go ahead and run step. And what I see that out of the over 4,000 genes now, 146 of those have a co-expression profiles based on the 3 gene IDs that I have used in my search. And so this is a really nice way how you can expand your search using not only current data sets that load in 56, but essentially any data that is loaded in PhoneGDB. And so moving on, I just also want to mention really quickly that we have loaded phenotypic data, and we normally try to manually curate 1 or 2 publications every release. And we also in this release have integrated a large scale data from functional genomic screening of Candida albicans. And I will click on the link to go to the dataset record page. So there's a lot of information available in terms of the different conditions that were used to analyze, but in summary, what the authors have done here, they have compiled, organized, and standardized Candida albicans functional genomic screening data from over 400 independent screens. And they generated a data library of genetic mutants and their phenotypes. And so you can mine this data now by using a search that we have developed for this data set. And as I mentioned before, the searches are always listed at the bottom of the page, but you can also bring this search into an action by essentially adding a step within search strategy. And so what I will do here, I would ask a question of how many of these genes that have non-synonymous mutations that have co-expression profiles in the calcin database also have some sort of phenotypic data from the functional genomic screening of Candida albicans genome. And so I'll click on the add step, and because I'm still searching for genes, I'm still working with the same type of data. I'll click on phenotype evidence, scroll down, and click on this search. And so as Amar have shown you, you can either select a fraction of the data, or you can either select all. In this case, I will click on the second option where they have used MOPS medium, which is media recommended for use with antibiotic gradient-based systems. And I'll say return any gene that have shown a phenotype when this media has inclusion of doxycycline. And so then I'll click on the run step. And here we go. So we essentially combined a comprehensive study of the SNP, transcriptomics data, and also phenotypic screen, and have identified 59 quite interesting possibly genes in Candida albicans that you may want to take a look if you're interested in antifungal resistance. So at this time, I will go back to the main screen, I guess, for the news. And we have about 1 minute left. So Amar, I'm happy to answer any questions if there are any.
I think we answered all the questions. I'm going to actually just take over and remind people of the different webinars But thanks very much. So of course, keep asking questions if you have questions, and we'll answer them. We had many during this webinar, which is great. We love getting these questions. So one thing I just wanted to remind you that, again, as you saw here, we have a number of webinars available, including, actually, let me go to our webinar page, and then this way you can see all of our help. So if you go under help, you can click on learn how to use VEuPathDB, and you'll notice we have a bunch of options for you here to choose from. Including, we have this calendar of events, which includes not only events that VEuPathDB is involved in, but also events that the second BRC, which I mentioned earlier in this webinar, is also involved in. And so you can scroll through the different dates and see what events the BRCs are involved in. The webinar page, I want to just point out to you here that we include upcoming webinars, and so this gets updated frequently. So for example, today's webinar is release 56, and you can expect, every 2 months to be a release webinar, which goes along with our news release to make sure everybody is familiar with what's going on. But the good thing is that all of these are recorded and which are available here. So if you were interested in getting a highlight of release 55, or if you're interested in learning about Go enrichment or any other tool that is available in VEuPathDB, you can find webinars that we ran over the last couple of years. These are all available, and they're actually heavily watched on YouTube, and so you can go to them and watch them. And so I would encourage you to take advantage of these resources. And as usual, feel free to contact us at any time using the Contact Us link. We love hearing from you. Let us know about data sets. Let us know about features that you think we should have. If you notice a bug on the sites, of course, we'd love to hear from you as well. And if you have general questions needing bioinformatics support, if we can help you, we're happy to help you. With that, thanks, Eve, Suzanne, and Gloria, and for your contributions. And we will see you next time. Thanks, everybody, for attending. Bye-bye.
Bye-bye, guys.
Bye.
Bye.